being at the end of term.
Okay. So hello, everyone. Welcome to the 2023 iGEM information session. I'm Melissa, a third year in microbiology, and I'm also the Vice President of Mentorship. I was a former co-lead of 2022, and I'm your host for today. Just a question to start us off. What's your year and your major? If you can annotate the screen or send us a message to all our participants. So we got some engineers, biologists, biochemists. So let's get started. So here are some, oops. Could our annotation people remove their annotations for us?
You can also clear them yourself, Alisa.
Oh,
there you go. Thank you.
Okay, so introducing some of our speakers. If you guys would like to state your role, your major, year of study, starting from Gustavo.
Sure. Hi, everyone. Can you listen to me clear? All good? Perfect. So, hey, everyone. I'm Gustavo. I'm the president of the UBCO iGEM club. And last year, I was co-leading the 2022 UBCO iGEM team with Alisa as well. So most of my duties were just taking care of that everything was going well, that we were in communication with all the prof. I was mostly in charge of wet lab and the section that is more in the laboratory. And yeah, basically all the big administration things along with Alisa.
All right. Josh, do you want to go next?
Yeah, my name's Josh. I'm in my third year of microbiology. I'm the former human practices lead for the 2022 iGEM team. And I decided to take a step back from the competitive side of things. I'm your VP of communications for the club, and I'll be acting as an iGEM mentor this year.
All right. And Shrishti?
Hi, I'm Shrishti. I'm in my third year of electrical engineering. I was the dry lab lead for the last year's competitive team. And this year I'm acting as the VP of engineering, as well as an iGEM mentor for this year's competitive team.
All right. So for our agenda for today, we're gonna firstly go over what is synthetic biology, what's iGEM, then we'll go with talk a little bit more about life bulb, a description of our subgroups and our leads, and then we'll talk about the competitive cycle, the application process, and then we'll have a Q&A session at the end. So Gustavo, you can take it from here.
Sure. So the basis, everything that has to do with iGEM is centered around synthetic biology. So I'm just gonna do a small explanation of what it is, because some people don't have a very good understanding of what it exactly is. Here I always say, what if we could save the coral reefs? What if we could smell extinct flowers? What if we could create new ways of making food that is better for everyone, that is harm-free? What if we could grow anything? And that is what we see in synthetic biology. So, next. The way that I define synthetic biology is redesigning life itself. So you probably heard about molecular biology. Here what we do is that we are using biological systems, cells, metabolic pathways, or other things that will probably or very likely never happen in nature or will take too much time to occur. And we are engineering them. So let's go to the next part. In synthetic biology, cells are biological machines. So we're seeing them as factories. DNA is the code, similar to the zeros and ones, the binary that you see in computers or in your phones. And the proteins, these machines, molecular machines, are the output. For evolution, what works, whatever is good enough. But if we apply our own design, our own understanding, we can start making conscious engineering and putting these two things together, engineering and biology. we start getting this new area of biotech that is called synthetic biology. So let's go to some more examples. Something that we like doing in this field is that we want to extrapolate. Probably some of you may recognize these symbols to my left, especially the ones in electrical engineering. Why? Because we use these symbols to kind of represent things that are going on in circuits. This thing is activating this thing, then we have some logic gates and other things going on. We started taking this concept from engineering in biology, in synthetic biology, to not have to think about the molecular interactions between every atom and every other thing. In this way, we are able to think above these things, just knowing how they will interact and make really complex syntaxes, really complex matrix of everything that is going on. We're going to have a more in-depth workshop one next semester about these parts so that you are able to understand and see how they work. But just have in mind that this is what we use in synthetic biology: promoters, coding regions, terminators. And all of this goes around the central dogma of molecular biology. Basically means that if we know what the gene is coding for, this is telling me do this, and this will create this protein, then we can go the other way around. This protein has come from somewhere else. So we're taking that, and using this concept, we can make really cool things. So let's see some cool examples here. Well, just to describe it more, synthetic bio. So this area started because we wanted to understand how biology works. Probably you heard about reverse engineering. So we don't think much about the molecular biology again, and we start taking all the parts. What makes a cell a cell? If you ask an engineer, for example, how you want to make a bridge or something like that, they probably want to know how the bridge works first than doing anything else. We need to understand how the parts work together to actually understand it. Most areas of biology, they don't really care about how they work, but in synthetic biology, biotechnology, bioengineering, we need to understand how these parts work. Our kind of ultimate goal is to be able to create a cell out of nowhere, to be able to put all the parts together and they should work. But to get to that, we need to understand every single part and start using them as Legos. So next. Here, the first objective was to understand the biological systems, what I was saying, reconstructing part by part. If I put this here, the other way around. If I take this and the organism doesn't work anymore, then probably it was really important. If I take this, is it still doing okay? Cool, we can take more and more. That's an area called minimal cells, but still really cool. But what we can do is the opposite. We can start taking these parts that we know what they do and start doing things that never happened before. We can start making design, we can start making order. And now I can tell you some examples. This is a genetic circuit. You'll learn in the future that basically what this does is that this is telling me produce GFP, green fluorescent protein. We use it for almost everything. It's green, it's fluorescent, and it's a protein. So let's keep going to the more complex things. You can do a lot of things in synthetic biology, basically. You can create really complex algorithms the same way that we have a phone, because there are many small parts that we know how they work. They're really simple, but put together, they can create tremendous things. So yeah, let's continue with more examples. This, going back to the genetic circuit, and here's what I wanted to talk about. Cool. So some examples. In nature, organisms, for example, have evolved ways of detecting heavy metals. Why? Because they want either get away from them because they can kill you, so we don't want to get close to them, or we want to get close to them because maybe we want them. Maybe we are a weird organism that is able to breathe uranium or whatever. So there's a thing that evolved that allows me to detect it. We can take that part and combine it with another part that tells us, for example, the green fluorescent protein. So now we know that if there's this heavy metal, it's going to be green. Cool. Now we can make that system really specific. We can high tune it so that it can detect really small amounts of this material. Examples where this will be useful, this is a biosensor. We can go to the Amazon. We don't have our devices. We don't have our spectrophotometers and other tools that we can use to measure heavy metals, because in this area, they do lots of mining. So there's a lot of contamination. So how do we know, for example, if the people there are in danger of being contaminated, of being intoxicated by these things? It's kind of really difficult because you have to send a sample to a lab. wait for that to go and all the stuff. You can test multiple samples if you use this technology. Basically, we engineer a cell, really tiny organisms, to have the ability of detecting metals, be green, tune it so that you are able to detect exactly what you're doing by just looking at it. Put it in a vial, you go to the Amazon, you take some sample of water, there you go, you check it, and now you're able to see it. Really cool tool enabled just with biology. You can do something similar with cancer. Cancer cells have special molecules that make them, that tells us that they are cancer. That's difficult to detect, for example, with a robot. That's hard because you have to get the robot all the way inside of your body. Or the other way, if you use drugs many times are not very specific. So some teams around the world are trying to use cells to actually tackle directly this problem. There are other teams that are trying to enhance the CO2 capture power of trees and plants, photosynthesis. We know how it works, but maybe it's not the most efficient way. Maybe we can get rid of this or improve it or change these little parts. Again, we are playing Legos here and we are using our own design, our own mind to improve these things. There's a really cool project that I'm gonna talk about later that is trying to use the power of smelling. I'm gonna go into that later, but yeah, smelling for a robot, that's hard. But for us, it's super easy. Why? Because of the way that we interact as biological beings. We can take these powers, again, look at it like powers in nature. We are taking these powers that all these weird organisms have, and we can match them, improve them, and modify them. Antifreeze proteins. There are animals in both sides of the planet that somehow live there when they should be frozen. So some of the techniques are having these proteins that are unable to freeze. So what we can do there is that we can take those proteins, we can put them, for example, in maize, so that it will grow even at really freezing temperatures. And we are doing that already. And we can even spray it. Some weird ideas that are going out there is, for example, concrete. Concrete, sometimes when it gets frozen, there's water inside. Water expands, it breaks it, and it's not good for anyone. So there's a group there that is trying to put antifreeze proteins in there so that it will not freeze. It avoids the ice crystals from forming, and that's amazing. And again, you probably heard about the plastic degrading enzymes. Basically, if you give it enough time to nature, they will be able to find a way of generating energy out of nowhere. There are bacteria there, some other organisms that are able to eat these plastics. We can take those machines and we can use them here. My message here is that many things that we can do, for example, with organic chemistry or with other techniques like physics or other things, are quite hard if you want to make really complex things. Life is really good at putting things together like atoms in really specific places, trying to go against the natural order of disorder. So that's what we're taking advantage in bio, and it's super cool. So yeah, next. Let's see.
All right, so I'm gonna be introducing iGEM. So does anyone know what iGEM is? Can you guys like tell me what iGEM stands for maybe in chat and what the competition is about? Yeah, it stands for the Internationally Genetically Engineered Machine Competition. Could someone maybe give me a brief description of what the competition exactly is? All right, so it looks like we are a bit unsure, but that's good because that's what this information session is about. So I'll just show you guys a video that was played at the Jamboree this year, which was in Paris. And this is like a video that kind of introduces like iGEM's history and what the traveling biobrick is. So it's an award given to grand prize finalists of iGEM. And so let's watch this video.
As we know, the grand prize winner takes home the traveling biobrick.
The grand prize for the 2006 iGEM competition goes to Slovenia.
winner of the traveling bio brick is University of Washington.
And for the 2014 grand prize in the undergraduate category, we would like Heidelberg to the stage.
The undergraduate grand prize winner, William and Mary.
Winner of the high school grand prize is
The undergraduate grand prize winner! And the grand prize winner of the undergraduate section is...
All right.
As we know,
There we go. So Josh, do you want to take it from here?
Yeah, of course. So with that segue, the International Genetically Engineered Machine Competition is an independent nonprofit that was started by MIT in 2003, aiming to promote the field of synthetic biology because it was really novel at the time and no one had really heard of it. And now over 350 teams compete from around the world to earn medals, gold, silver, bronze, as well as special prizes in a variety of categories from energy to health-related therapeutics, diagnostics, and even environment or climate change projects. And they're all aiming to tackle issues seen on either the local level or on global scales. And it's usually been held in Boston, but the competition itself has gotten so large that they needed to relocate to Paris, which, we're not complaining, obviously. And that saw over 4,000 participants this year.
All right, so a bit more about like the Jamboree, which is like this huge venue where all the participants, including startups, experts from around the world, prominent in biofigures, and then biotechnology representatives such as Promega and Ginkgo Bioworks all come together. It was held in Porte de Versailles of Paris in October this year from the 26th to 29th. And it was like a really good opportunity for networking, meeting like-minded students from across the globe, obtaining feedback for our projects from diverse experts, especially in the case where we're considering like a startup. And so this year, our team won a gold medal and we were nominated for the best wiki for our project Lifebulb. It's basically a sustainable alternative towards modern electrical lighting using a carbon negative system in cyanobacteria. We basically envisioned it to be used in a variety of situations such as architectural purposes, indoor decorative lighting, and a medium for art expression, collaborating with Les Louise, a indigenous Silex artist, to create the concept art of Lifebulb. So I'll just show you guys a quick video that we did, a promo video for our project.
Imagine a world without light, a world without its vibrant radiance enriching our lives, a world without our illuminated streets and homes. Electrical light is everywhere. We depend on it for every aspect of modern life. However, the development of light has a dark side, a problem that nature has never faced. High energy consumption, greenhouse gas emissions, and other environmental impacts. But what if a solution already exists in our natural world? Throughout history, we've been fascinated by the mystical glow of life. Indigenous communities all over the world have used and told legends of this natural phenomenon called bioluminescence, nature's ability to produce light. We at UBCO iGEM are harnessing this natural occurrence to grow living lights that can be scaled up easily, clean our air, and be powered by the sun. To do this, we are engineering photosynthetic bacteria to bioluminesce. We will express the bioluminescent system of fungi in these cyanobacteria, producing a soft green glow while converting greenhouse gases into oxygen. Our light will adorn our parks and decorate our homes, providing a serene space free from the distractions of electrical light. Let's use this new sustainable alternative to reconcile with nature, to learn from those who were here first, and to reduce global carbon levels while providing an educational experience for the public, a profound medium for artistic expression, and an opportunity to demonstrate the beauty of synthetic biology. For a greener future, illuminated by life, a life ball.
All right. And so what kind of awards can you win at the iGEM competition? So we have, as Josh said, 16 special prizes awarded to like the most innovative and unique teams for certain categories. We were personally nominated for Best Wiki due to our design, graphics, our comprehensive information, and the documentation of the process. And in addition, there's track awards. So every team chooses a track that iGEM believes is the most significant challenge in our society. And iGEM teams with the best projects in each track get a track award. And so on top of that, there's also medals, so gold, silver, and bronze. So gold is awarded to those shown excellence in multiple criteria above and beyond the key essentials of an iGEM project. For silver, they need to demonstrate and fulfill the key pillars of the iGEM project. And for bronze, this is more of the participation in making a difference for future iGEM teams and fulfilling the bronze medal criteria. So to earn these medals, you have to convince the judges. And on top of that, over 350 teams, there's the top 10 in the undergrad, high school, and overgrad categories. And then the grand prize winner is selected from each category. So what is an iGEM project? Is Gustavo able to take over here, or?
Let me see. Can you listen to me all good?
Yep.
Cool. Okay. So an iGEM project. This is kind of similar and also different to a research project that you'll do in the lab. So what we do here is that we take an idea. Our objective is to try to solve a problem, try to do something different, try to use synthetic biology, what I just explained, in a creative and new way, something that we're doing for the first time. So the different thing here is that we are aiming to have an incredibly multidisciplinary group. So not just scientists, not just biologists. We want people from all points of view so that we can integrate these things into a project that is full, that is complete, that is well-rounded. So we integrate a section that we're going to go through them in a bit called wet lab. That is everything that we do in the lab. Then dry lab, all the sections that we have to go above that. Then we have something very particular. It's called human practices. Josh will help us with that later. And what we do is that we try to take this knowledge, these new ideas, and try to solve these problems using biological tools that we were seeing before. So taking inspiration from all these issues, how we can solve them, and from our own perspective. So let's see some examples. There we go, cool. So I really like to talk about these examples because I think they're amazing. Usually what happens is that the team comes up with a problem or some inspiration. And let's say, I really want to solve this problem. I think that that's something that we don't have a really good solution, and perhaps biology can solve it. Perhaps there's a better way. The nice thing about biology is that if you let it, the nice thing about biology is that it can, which means that you can make it really big, you can make lots of it really cheap eventually. So let's talk with some of them. I was for the topic about a robot that can smell. So that was a project from Zurich, iGEM 2018, Aroma. What they did is that they found, they were asking themselves, can we make a robot that can smell? Can we do something like that? So what they did is that they engineered E. coli or other bacteria to sense these molecules. again, they have evolved the ability to sense these molecules, and then you can take that and combine it with something else. So what they did is that they were able to, for example, make the bacteria smell to lemon, lemonene, and other molecules, terpenes. So that they were able to then send a signal, either electrons or something else. I'm not sure what they used there. And then they connected that to a hardware, to actually something that was able to detect that and tell the robot X, Y, Z coordinates. Sure. So all these different things. And they were able to make really cool things. A robot that can smell with biology. Morphe, they look at the problem of dyes. So the clothes that we use everywhere else, the production of color is very pollutant. So what they did is that they look in nature, something called biomimicry. How can we produce color in a more eco-friendly way? And they found the structural color. They modify the biofilms that bacteria naturally produce to try to get colors that are eco-friendly. Really amazing. And this year, the winner of the overgrad team was Netlantis, something amazing. So they all came from, that was a nice thing. All the team came from coastal cities. They were all very passionate about the sea, and it was really important for them. So they were saying, what can we do to try to help the oceans? One big problem is that we fish a lot, and that has some detrimental effects on the fauna. Why? Because the nets usually trap other things that are not supposed to be trapped. They end up as trash in the ocean, and that's not good. So they were asking, can we make some environmentally friendly fishing nets? They took the genes and the proteins from two different spiders, they put it together, then they did assays to try to see what was the strength of it, and that they kind of didn't work. Then they found that there was this marine spider, they took the genes from it, they put it in there, it worked. They designed a digital simulation of how these proteins interact, and they were able to see that the way that they were spinning the fiber could be improved. So they integrated engineering concepts, computational concepts, mathematical concepts, and the biological ones. I really suggest you, if you have time later, we can provide you the links. Just go and look for their promotional videos and their presentation videos. They are also in the iGEM Jamborees. Amazing projects, taking inspiration from nature to try to solve these problems. Really cool. Next. So, as I was mentioning, one of the big components, I wouldn't say the most important, but definitely the one that does the physical part of the project is the wet lab. Does anyone know what wet lab is? Just really quickly, I want to know. You can raise your hand if you have any idea. Just want to see if anyone knows. Probably not. Okay.
I know
so
at
let's
least
go.
one of us knows.
Why don't you tell us?
Okay. Are you gonna wait for that? No? No. Okay. If you're typing, we'll continue. So next. So if I were to describe wet lab, is what will be the lab work. So you can see some pictures of us working in the lab here. Basically, we do all the things that are genetic engineering, all the parts that involve us getting into the lab and doing molecular biology, microbiology, doing some kind of tests. Everything that is that, we do it here. For these projects, what we do is that we design first our system. We go through the engineering cycle. I believe we're going to talk about that later. But the first part is design. So we design the strategy, our genes, and then we have to put them into reality. We have to synthesize them, and we have to here cut them and paste them in a molecular way so that we have everything together. That's the building section. Then we have to test them. We have to do certain assays. We have to run gels. We have to do all sorts of different things. So let's go to the next slide. Here are some of the techniques that we used last year. I have a couple more that I just remembered. But just to let you know, we extracted plasmids, we did gel electrophoresis, we PCR, we did Golden Gate, SDS page, inoculation, plating, cryostocking, and other things. So these are techniques that you'll probably start learning by the third or fourth year of your degree if you are in sciences. If you're not even in sciences, you probably never learn about them. So that's something that our club, separate from the competition, we are trying to help you to provide these opportunities. But in the iGEM team, if you're in wet lab, you'll be able to learn about these things and many others. The idea is that every project is different. Every project has its own components. So for this one of the things that we wanted to try, but we didn't have time, was using an HPLC. I believe none of us ever used a HPLC before. And the technique is to go to the prof, in this case, Dr. Dang, Dr. Mahmoud, Dr. John, and ask them for help. They're going to be our advisors. So they are going to help us do these techniques, learn how to do them, and we are the ones doing everything else. Something that is different for iGEM is that this is a student-led project. These projects are things that you will probably have a hard time funding if you were a researcher. What you usually do if you go to a URA or other possibilities of doing research here is that you have to adapt to whatever is already on campus. It's an opportunity for you to start learning these new techniques and do something new. This was the first time that someone was doing genetic engineering on cyanobacteria in this campus. So, yes, students, really cool. Next. Some of the other things that we do here have to do with genetic engineering and design. So we also use some software, BenchLink here, for example, we literally type DNA. I have people in my team that were already had to learn how to do this. They started from almost zero and now they are able to, we need to add a promoter, we need to add this thing, we need to cut these things. We can run these things online in silico and try to do all these different things. Really cool. We use all sorts of different kits that we get from different companies. We start doing the simulations. We got our parts, we have to pick them up. It's literally your opportunity to feel like a researcher. You are in the lab, you are in charge of your experiments, and you have to be self-directed. You are the one directing the entire thing. Which experiments you do is your decision. How you made them, your decision. How to learn about them, your decision. We have all the resources. Even if you don't have them here on campus, we can always contact people that are abroad. We have done that in the team before, so with an expert, for example, in Germany, about the things that we're doing. It's a really big thing. Amazing. I don't know if anyone wants to add something else to wet lab. Nope. Oh yeah. Okay.
So as Gustavo kind of mentioned previously, dry lab is everything that is not done in a lab. So it's the analysis of lab results or hardware or software to do with the lab results. And there's three main components to dry lab. I want to go to the next slide. The first component is mathematical modeling. So iGEM defines this as deterministic exploratory molecular dynamic and or stochastic models. It's exploring the physical modeling of a single component within a system and utilizing mathematical modeling for predicting function of a more complex device. So the way iGEM teams have commonly interpreted this have been enzyme kinetics modeling, metabolic modeling for optimization, or also financial modeling. So I'm just going to go through a couple of examples. The first one is Eindhoven in 2022. I believe they won the special award for best model with their model. They made a kinetic model about the signaling pathway of their engineered cell to simulate the generalized extracellular molecule sensor receptor and its activation. So this model was designed to simulate the behavior of the system in normal and in extreme conditions by only changing certain variables. This way, the system's behavior can be predicted, and the model improves and reduces future lab work by predicting optimizing conditions and useful experiments. So it acted as an aid for their lab work. It didn't substitute it. And then Patras, which is the next example, I believe, or no, it was Tepe in 2021. So they also won the best model for their year. And they made 3 different models actually. And one of the models was a financial supply and demand model. So their model was aiming to predict the incidence of blood shortages by blood type under a variety of conditions. So their project was able to convert O to O type blood in case of shortages. So this data helped them predict and refine the implementation of their project on a greater scale. So the definition for a model is really broad, and there's been a few interpretations by iGEM teams in the past. The second component of hardware, of dry lab is hardware. So hardware can be an implementation of your project. It can be a modified existing piece of hardware that can be improved or changed due to your project. It can be hardware that contributes to making your project run or work, and it can be made as a physical prototype or modeled on software. So for this year's project, we modeled it on software. I modeled it on SolidWorks, but teams in the past have also made a physical prototype. I know we saw a few in the biomanufacturing track where they had made a physical prototype and brought it to the competition. The third component of dry lab is software. Software can be implementation of your project. It can be created as an aid for mathematical modeling. It can be a dynamic database. It can be software that's integrated into your hardware. This year, we did make a small software for appending prefixes and suffixes for DNA strands for promoters and various parts of that genetic circuit that Gustavo mentioned. We also have that on our wiki.
All right, now moving on to human practices. Human practices is a term that's rather unique to iGEM, and it involves exploring the legal, the ethical, social, the economical influences of our project and what they will have on the world. And so that means doing like educational outreach and interviewing field experts for their unique perspectives, and just generally speaking, integrating their feedback and developing a story surrounding your project. And another huge aspect is educating the public on GMOs, because there's a stigma in that regard, and just generally improving the synthetic biology knowledge in your community, putting on workshops for students and other educational, creating other educational resources, and then also partnering with other iGEM teams through a variety of collaborations and major partnerships within the competition. So just to begin with human practices, it often starts with the background research. So that involves, first of all, defining your problem and what is it you're trying to solve? Is it a local issue? Is it something that has an effect worldwide? And then determining an appropriate solution. Is that solution actually good for the world? Is it actually going to have a positive impact? And so when you're designing your project, you have to consider which values you're trying to target. Are they environmental, social, moral, scientific, of course? And then branching from there, which communities are you looking to consult with, and which resources are you trying to, do you need to look into? So with that comes proposed implementation. You have to determine what are your end users. In the case of Lifebulb, we ended up creating several models, an architectural model, as well as a consumer model. So in that case, we were targeting our products towards children and teens to educate them in synthetic biology and create some sort of lamp that had an artistic capacity and that was novel in many ways. And so, yeah, determining how your project is implemented and then considering the safety considerations. Biosafety and biosecurity are huge aspects to iGEM and human practices. One has to consider, first of all, the organism you're working with, which risk group is it? Does it have any toxic secondary metabolites or any intermediates within the pathways that are going to cause harm? What sorts of lab procedures are you going to have to do to mitigate that? Things like that. And then with integrated human practices, you're actually taking the responses from the people who you've consulted with, your stakeholders, and reflecting on them, integrating their feedback back into your project, and altering the purpose, the design, or the execution. And so here's just a few examples of our integrated human practices, some of the work we did in our community outreach. We spoke with lighting designers, we spoke with Indigenous artists, with bio artists. You see me there, I think I was messing with our logo in the design process. We talked to bioluminescent companies. You do a lot of design, so I was working with our hardware team for our proposed implementation, and then creating our promotional video. There's a lot of design aspects and marketing aspects there. And then moving on, another aspect of human practices, as I mentioned, the collaborations. This is pretty broad, but it's a competition requirement that can involve possibly mentoring other teams. For example, UBC Vancouver was our quote unquote mentor this year because they are our sister campus and we're our first year team. Other teams have just shared protocols in the past, built models together, troubleshooted their projects. I've seen teams do, they've created workshops together, they've created like children's books, all sorts of things related to synthetic biology. And then a partnership is something more significant. For example, we collaborated with Arizona State University this year in a bunch of recurring meetups to develop a handbook for phototroph symbio, and also host a community of teams working with plants, microalgae, and cyanobacteria. You can learn a little bit more about that on our wiki if you're interested. And then as for the education and communication, as I mentioned, building workshops and materials, presenting at events across the community and on campus, and the scientific communication to support the implementation. So there's the handbook cover, as I mentioned, some of the meetups we had with a variety of teams, from small collaborations to large ones, some of the events that we hosted on campus, and for students K through 12 throughout the Okanagan. We created a manual for Life Bulb on how to use it, should it become a reality. We spoke with media to present our projects, and we also presented both to the judges in a judging session and to the public at the Jamboree.
All right, so finally, our finance subgroup is basically the backbone of all our research and our iGEM team. So as part of finance, you'll need to think outside the box and of who we can possibly reach out to. In the end, we need approximately $25,000. So this includes not only filling out sponsorship forms, but also setting up meetings with investors and petitioning for their support. You'll also be planning events to fundraise on and off campus, usually in tandem with other clubs on campus. So you'll need to be able to build and nourish these results to reach as many students as possible. Overall, finance requires that you not only handle money responsibly, but also have great persuasion and people skills. So this makes finance one of the most versatile subgroups on our competitive team. For example, finance this year was able to secure 15 different sponsors for our team just in the span of less than a year. This was an incredible feat since considering we had no reputation as a competitive team at this point and had hardly any recognition on campus. So it's a lot different from where we are at now. We also started a GoFundMe and then planned some events with other clubs on campus as well. So as for the competition cycle, this is like just a typical iGEM timeline. We obviously are in like the off season right now, November to January, just reflecting on the previous iGEM projects and then just recruiting new members. In phase one, we start our new projects. So we brainstorm ideas, finalize the teams, the principal investigators, and advisors, and then we begin fundraising initiatives. And then in phase two, that's when we start all the dry lab, wet lab, human practice initiatives. We work on our wiki page, and then we collaborate a lot with other iGEM teams. So in phase three, we have between September to October, we finally prepare our project video, we build and practice the final presentation, and we wrap up all our projects. And then finally, we'll be able to attend the Grand Jamboree in Paris. So here's another breakdown of the process. This was on the official iGEM page. The months are a little bit different from how we go about it, but the basic main progress checks are correct. So we start with the team and begin fundraising, we brainstorm, read obviously the competition requirements, design, test, build, and etc. As for our wiki, we just want to show you some examples. We can start with implementation if Srishti wants to take it from here.
Yeah, so your wiki is basically supposed to be a well-documented website of your work over these 8 months. So just as an example, this is our wiki. This is our implementation page. As you can see, we have the drawings from Les Lewis, as well as the interlap model that Alyssa made. Yeah, this was just detailing the steps that we took for implementation. This is our Meet the Team page, which we spent way too long working on. It has every single team member and kind of their major, their interests. So we tried to make this one as fun as possible. We made a little video for it. This was the team that won Best Wiki in 2020, I believe, as well as the Grand Jamboree Prize. This was Lithuania. Their team is incredibly artistically talented. And this is kind of our gold standard for wiki when we were designing our wiki this year. This is, we wanted our wiki to look as good as this because their wiki is amazing. Another example is the 2021 Calgary team, Neocycle. They also had an incredible wiki. So what the judges are basically looking for is an aesthetically pleasing and functional wiki, as well as just well-documented pages. So a lot of wikis that we saw had really bare bones kind of pages with not a lot of information on there. And we really tried to steer away from that and pack as much stuff as we could, because that's ultimately what they're judging us on, is the wiki and all the stuff that's in there, more than the presentation and the judging session itself. I don't know if you guys have anything to add to that.
For our team, we stayed up around 24 hours for coding our wiki right before the deadline. So it's a very intensive process, but everything it's put on that wiki. So it's very high stakes. So that's how much time we needed to dedicate to that. So as for the application process, applications are currently open right now. They'll close and they're due December 22nd. So emails will be sent out to those who receive an interview. And then between January 9th to 15th, we'll be booking their interview, or they'll be able to book their select times, and then we'll be interviewing them. And then after January 15th, we'll send the email to those who are accepted. So please make sure to keep an eye on Discord announcements or Instagram stories, because that's where we'll be announcing to check your emails and things like that. So what exactly are we looking for when you are applying? So first of all, proactiveness. We're looking for several characteristics such as proactiveness, organization and time management, and accountability. So this involves learning outside of the work time, going above and beyond, putting this iGEM as your #1 extracurricular, and then keeping up with tasks assigned by leads and showing up to meetings. So for us personally, over those who have a lot of experience, we value those who are able to dedicate their time and get things accomplished independently without having to be taught. So we'll be looking for these traits in the applications and the interview. So it's very important that you write thoughtful responses to demonstrate who you truly are and why we need you on this team. Do you guys have any other things to add on what you're personally looking for?
I mean, yeah, for human practices, for example, I'd say it requires a very holistic view of the project. I constantly had to work with and understand the concepts of dry lab and wet lab, and then communicate their work back to stakeholders. So someone that's passionate and responsible is definitely a must there. And then also being just like socially conscious and creative in thought, because building your human practices is very unique to your own project. No one team has the same human practices experience. And it's really about drafting your own story. So someone who's creative in that sense is also really important, I would say.
All right, so what do you gain from this? So it's an opportunity to enhance your research skills and technical skills. You can experience leading, self-directing, and communicating as a team in a very intensive, large-scale project. And you also gain a family of like-minded, hardworking students. So this is different from other typical research positions because of how multidisciplinary the project is. And it's guaranteed that you won't be focusing on just one specific group or area, but you'll be developing a diverse skill set from being a part of so many different aspects and fields on basically a professional level. So for our own experience, do you guys want to answer what kind of stuff did we learn and what you guys are most proud of? Josh?
Yeah, so beyond HP, I also helped out in the lab on occasion. So I learned lab skills in molecular and microbiology. So developing a lot of research skills too around the initial ideation phase of our project, and improved upon my communication. That's a huge aspect that you can take anywhere.
Interesting.
Yeah, I think I learned a lot of synbio. Like I had no bio experience before this. And even just through dry lab research and work, I learned a lot about synthetic biology. And also I learned a lot about HTML from coding our wiki, which is the thing that I'm most proud of.
Yeah, you gain a lot of skills that you can apply towards like career or other extracurriculars, not just like iGEM. All right, so we do have like a short video of our personal experience and our memories that I wanted to show you guys. It's pretty funny. So it's like a thing of all the fun, nice, exciting videos that we took over the competition season.
When I gave the word, now in the morning I sleep alone, sweep the streets I used to own. I missionaries in a foreign field. For some reason I can't explain, I know St. Peter won't call my name. Never an honest word, but that was when I ruled the world. That was when I ruled the world.
Okay, it's recording.
Oh, jumping. Jumping. What? You get a burger, you're Christian.
Can we all just scooch forward like a little bit? We're not against the ball. Perfect. Okay, ready? Perfect. Yes. Are we ready? Yeah. 3, 2, 1.
That was when I ruled the world.
All right. So that was kind of a summary of iGEM and our experience. So now we have a Q&A session for you guys. If you guys have any questions, this is your time to ask.
You can send something to the chat or just raise your hand and ask. Be happy to answer.
You can also text us on Instagram after if you think of anything after the meeting. And this recording is going to be available to watch on our Instagram shortly, I believe.
Yeah, that's right. Send us a message on Discord, Instagram. You can email us if you have any questions regarding the application or just generally about iGEM. We'll be happy to answer.
All right. So if we have no more questions, I guess we can end the information session. Thanks for showing up, guys. So obviously at our club, we like to end off with the question, how will you change the world with synthetic biology?
Thank you, guys. Alyssa, did you just want to send the links to Discord or to the application? I'm sure they probably are.
I'm going to stop the recording.
Okay.